Good morning, everyone. My report is all about the process of meiosis. But before that, we need to know what is the use of meiosis. The reproduction of most organisms is when two gametes that each came from each parent is to unite, forming an embryo. This is where meiosis happens, the forming of gametes. Cells that undergoes meiosis is called the germline cells. In diploid organism, the germline cell has two copies of each chromosome. After the germline cell goes through meiosis, it became haploid gametes, which is the sperm cell and the egg cell. Before the germline cell begins the process of meiosis, it duplicates its DNA during the S phase, here. And this will be now the new germline cell. New duplicated chrome is called sister chromatid. Now, meiosis begins. There are two cell division during meiosis. The first division is called meiosis 1, here. And then, followed by meiosis 2. During meiosis 1, it starts with prophase 1, where duplicated sister chromatid at the centromere forming pair. Each pair now undergoes synapsis to form an exchange chromosome material, which this process is called recombination or crossing over, making the sister chromatid not identical to each other. While in the nucleus, the nuclear membrane begins to break down, and then the centrosome relocate to the ends of the cell, randomly attaching their microtubules to the pair of chromosome. After prophase 1 is metaphase 1, where the synapse chromosome in the equator of the cell is then proceeds to anaphase 1, wherein molecules chromosome separates at two poles. The cell with its telophase 1 cytokinesis, as they pull the cell, it is divided into two daughter cells. Then proceeds to anaphase 1, wherein molecules chromosome separates at two poles of the cell. This is called telophase 1 and cytokinesis. As they pull the cell, it is divided into two daughter cells. This two daughter cell now begins to meiosis 2, where it starts at prophase 2. Prophase 2 is the same as prophase 1, but there is no recombination because there is only one copy of chromosome. Next is metaphase 2, starts by aligning the chromosome at the equator of the cell randomly. Then anaphase 2, where the sister chromatids are pulled apart as microtubules separate, pushing two sides of the cell, causing telophase 2, wherein nuclear membrane reform and cytoplasm is divided into two haploid daughter cells. This division is called cytokinesis, and these four haploid cells that are created are called gametes. Thank you.